Welcome to the IndieBio virtual space. My name is Julie Wolf, and I am part of the new team here in New York with our new New York IndieBio program. We are going to have a really lively conversation around the future of genomics in medicine with two founders who are themselves using some of this technology to bring it to market and really change the way that we interface with our genetic information as we make decisions for our health. I would first like to introduce Joseph Pickrell from Gencove. Thank you, Joe, for taking time to speak with us today.
Yeah, thanks a lot for having me. Pleasure to be here.
Thank you very much. And I would also like to introduce Toby Call, co-founder of Chronomics. And not only would I like to say thank you for taking time to speak with us today, Toby, but also thank you for staying up quite late, because it is, I imagine, a little different of a happy hour situation when you're over in the UK, which is where Chronomics is. So thank you.
Like an evening coffee to wake up. My pleasure to be here.
Evening coffee, excellent. Great. So I've given your names and your companies, but I think that our audience would like to hear a little bit more about your scientific background and the companies at which you are using these. And so I wonder if you could each just give a little brief introduction to your background, your motivation to start a company, and what your company does. And Joe, I'll turn it over to you first.
Yeah, sure. So I am a statistical geneticist by training. So did my PhD at the University of Chicago in human genetics, and then did a postdoc at Harvard Medical School in population genetics, working with David Reich on how populations of using genetics to understand how populations moved around the world. And then I was a professor at the New York Genome Center in Columbia here in New York, working on statistical methods in various applications in genomics. And while this was purely in an academic type setting, what we became interested in was how we could use software tools and the great sequencing technologies, sequencing hardware that was coming out to really make these types of technologies industrial scale. And so at the New York Genome Center, we had all the latest and greatest Illumina machines that could do tens of thousands of genomes a year at $1,000 genome. And we were thinking about ways of how could we instead be thinking about doing millions of genomes a year at more like $10 a genome. And so how can we take this, the throughput and the price point of genomics to something that's really industrial scale. And so the technology that we settled on is one called low-pass sequencing, where we spun out the company, and that's what we work on today. So a little over 30 seconds, but thanks.
Oh no, that was great. And don't worry, we will get into low-pass sequencing and what that means in just a second. But Toby, I wonder if you could do the same for your story and the story of Chronomics.
Absolutely, yeah. So my background is in, firstly I did a biological science at Oxford University, and then ended up doing synthetic biology as a master's in UCL, and then moved to Cambridge for my PhD in industrial biotechnology. And as opposed to Joe, I actually don't have a genetics background. I was more in bioelectrochemistry and bioenergy sort of world. But it's at Cambridge where I met my co-founders for Chronomics, and one of my really strong motivations for that is the prevention of chronic diseases. I have an autoimmune condition myself, and that experience plus really being able to see what my close friends were working on in the epigenetics of aging, and more kind of in more detail in the confluence of machine learning and DNA methylation analysis, really kind of blew my mind as to what was possible and what would be possible, make possible with genomics over and above genetics. So at the moment, probably the best way you could sum up what Chronomics is serving the world as a kind of biodata API, specializing in making complex data available to use, and ideally in the medical setting as well. And fast forward to COVID times, we are doing COVID PCR testing, and are really excited as to how that is accelerating a new wave of sort of distributed precision healthcare.
Yeah, absolutely. Yeah, that's an exciting development for Chronomics. But I do want this to be a fun conversation. I prepped both of you by saying this is the title of our event, How Will Genomics Change Medicine? This is such a broad area. I'm sure we can't even cover like a 10th or 100th of all the ways that genomics will change and are changing medicine. But I would like to ask you first to address this really large question. It's kind of a fun way to kick off the conversation before we get into the technologies that you both talked about, the low-pass genomics and the epigenetics that I also want to cover. So whichever of you feel like you have an answer, and I will give a couple other examples of how you might take this, what direction you might take it, because genomics technologies are changing in a way that are going to change our way to predict things. They will change accessibility. There's a number of different ways that you could imagine the future of medicine using this type of data.
Absolutely. Maybe I'll just jump in with a report that the UK government just released last week, actually, called Genome UK, which is a pretty strong statement about the government's sort of ambitions to really include genomics as a core part of healthcare in the UK, which is very exciting, really, to see a system, you know, as big as the NHS looking forward. But yeah, you mentioned some of the key issues. I mean, you know, in terms of, you know, well, I guess the goals, you know, would be around, you know, precision medicine, of course, diagnostics, accurate diagnosis, precision treatment pathways, and then through to prevention, early screening, and things like that. And I think one of the major barriers is, you mentioned, access and costs. And it's really exciting seeing, you know, companies like Gencove actually make those $100 potentially, you know, below that a reality, because that, you know, is a barrier to a lot of applications that could happen. So that's really cool. I mean, I'd love to hear more about Gencove's technology, as mentioned, Joe. So, but yeah, it does seem to me, you know, that the biggest opportunities with medicine, you know, more accurate diagnostics and sort of preventive screening in the future. And then obviously, I'm biased, but I'll say, you know, moving beyond to epigenomics and the other wonders of the genome.
Yes,
Yeah, I was also the UK government report, which I thought kind of covered all the bases. The UK government has been really like blazing a trail over the past 10, 20, 30 years in how genomics is used both clinically and in research. And so if you look at the report, you know, honestly, I wouldn't have too much more to add. Like, they covered all the bases. Like the top applications in diagnostics are things like cancer, early disease screening, pharmacogenomics, and then moving upstream to sort of population screening and sort of moving genomics into the space before someone's really sick, in the space of population screening and estimating disease risk in new and creative ways. And I think those are, I think the other thing they mentioned is research, which of course is extremely important to drive both of those. But those two types of applications are, it's great to see a national government think about the things at that major scale.
Yes, absolutely. So why don't we dive into some of the weeds of the technologies that will drive those applications. Why don't we start with you, Joe, and low-pass genomics, which is the core technology of Gencove. Can you give maybe the low-pass genomics 101 summary?
Yeah, absolutely. And so a lot of the reason we were working on this at the New York Genome Center was, again, we had all this sequencing hardware, which had particular throughputs and particular cost prices associated with it. And what we were thinking was, how could you dramatically increase throughput and dramatically decrease cost? And the approach that we took is one where, instead of doing what's called a deep whole genome sequence, where you measure the entire genome, call it 30 times on every individual person to get a really accurate picture of their genome, and you run a sequencer on one person or 10 people at a time or whatever it is, our idea was load as many people onto a sequencer at once as you possibly can fit, get a little bit of data on a person, and then use the existing data that's out there to sort of fill in anything that you missed. And so what we know is that we can design machine learning algorithms, things called genotype imputation algorithms, that take advantage of the fact that if I measure somebody's genome and I look at position number 1, we know that if somebody has a particular base at position number 1, we can look at nearby positions 2, 3, 4, and 5, and they're correlated. And so we can sort of fill in the blanks, and using the data that's in the public domain or that we acquire, we can use that correlation structure to dramatically decrease the amount of sequencing that we need to do in an individual person, which drives down costs and makes us really in line with the mission of Gencove, is to make genome sequencing technologies very accessible, sort of widely available across industries, and interpretable. And so we provide that software layer to make that happen and really drive up throughput and drive down cost of genome sequencing.
So it strikes me that this type of technology is really dependent on having that database of genomes. And that's something that we can talk about, but is that an accurate statement that low-pass sequencing is only possible because of the large number of genomes, whole genome sequences that are now in repositories around the world?
Yep, absolutely, absolutely.
And let me ask a follow-up question, because you mentioned pharmacogenomics as covered in that report, and that's something that is also taking off. It's looking at various loci in order to determine how someone might react with a certain medication, for example, right? How they may metabolize it, or how much their dosage may be personalized, right? That's really leading to personalized medicine. Now, how would low-pass genomics how it compare to something like genotyping for that type of service, right? So you could probably intuit based on these short reads that you get, and you can say, oh, well, we guess this or guess that based on statistics, versus actually sequencing some of those reads.
Yeah, and so the way we've thought about this is the primary use case for low pass whole genome sequencing in this type of analysis is in the discovery phase of. So now imagine you have a clinical trial of tens or hundreds of thousands of people, and you want to identify genetic variants that are predictive of whether someone responds to a drug or not. What's the right technology there? And what we've been able to show is that low-pass sequencing, compared to some of the other technologies that are out there, like a genotyping array or whole genome sequencing, allows you to really just scale those sorts of studies up on a fixed budget. So you have a fixed budget, you can do tens of thousands of people rather than hundreds or thousands of people.
So the big advantage would definitely be cost, and I imagine that you must also validate, right, to make sure that the same types of data come out of both of those technologies, correct?
Yep, exactly. And so we've done a number of head-to-head type comparisons here. We have a preprint on bioRxiv in collaboration with a group at GSK out in the UK, looking at these exact types of questions. What
about when you get all of these samples from various patients? Do you have to prep them in certain ways that vary from, let's say, whole genome sequencing or other types of sequencing technologies? Yeah, so we've worked on really what the way we've approached this is, again, to sort of think about how to take it to really industrial type scale. And so a lot of the things we've done on the lab side around automation and miniaturization and turning this into the type of solution that can be easily scaled up to not tens of people or hundreds of people, but hundreds of thousands or millions of people. And so there's some work on the lab side to really enable that. I see. And I would guess that it's kind of sequencing technology agnostic. The important thing is just getting accurate data, whether you're using Nanopore or Illumina or whatever it is that you eventually apply. Yeah, and we see sort of, if you look out in a few years, maybe that the actual sequencing technology or sequencing data itself has become somewhat commoditized in the sense of it doesn't matter whether it was generated on an Illumina machine or a BGI machine or by a Nanopore. There will be, of course, some important differences between the different technologies, but a lot of what's important will then be, once you generate the data, what do you do with it? And so how do you interpret it? How do you turn that into something that's useful? Right, and how does the end user use that to make a decision, right? And we'll get into that in just a second, but that's, I think, a pretty good overview of low-pass genomics, excuse me. Very interesting, and I'm gonna turn it now to you, Toby, to talk a little bit about what epigenetics is and how it even fits into a discussion on genomics, which of course, for many years, people were just thinking about whole genome sequencing, and now we know there's so many more layers to it.
Yeah, absolutely. But just first a comment on what Joe said about democratizing sequencing data. But that's definitely going to be the case. You could see these companies becoming like they're just the desktop providers, for the same kind of computing that we'll do on any computer, but it doesn't matter what source it's coming from. It's data going into the right pipelines. But Yeah, so with epigenetics, actually we have a sort of similar parallel story talking about sequencing and why we champion sequencing over potentially other methods like arrays. Although we're looking at epigenetic changes, so our pipeline is based on bisulfite sequencing to highlight where epigenomic changes are happening due to DNA methylation at CpG sites. And one of the really big advantages of sequencing in our approach is that it allows us to see a much broader view of the genome and take into account many more sites than you could do with other technologies like arrays. And off the back of that, you can then do look at sort of much more targeted panels once you've identified those sites of interest, for example, correlating to longitudinal changes in phenotypes, that kind of thing, and then drive the cost down. So sort of similar to, well, in a different route, but producing lower cost biomarkers from the genetic level, but obviously looking at longitudinal change. And then to kind of, I guess, backtrack a bit in terms of why we think epigenetics is important, but it's, you can think about the components of risk for disease, and of course, genetic risks for disease are very real. But when you look at the major non-communicable diseases, the components of risk due to genetics can be several times, 10, 20 times potentially. When you look at lifestyle and environmental factors, cumulative life impacts, you're looking at effect size of potentially thousands. It's much more of a story, and those are with epigenomic, if that makes sense.
Yeah, that does make sense. I think we lost you just at the end, but just when you were saying, does that make sense? So I think we got that. And actually, I would like to ask you a little bit about this, because it seems to me that epigenetics and transcriptomics kind of go hand in hand. So I wonder if you could talk a little bit about how it would be, I don't know, different or more advantageous to look at the epigenome and where those methylation sites are, rather than, I don't know, maybe it costs more, or maybe because RNA is more labile, to look at the transcriptome of patients for similar reasons, right? Like to see what is being expressed.
Right, exactly. Yeah, I mean, there's even, there's really interesting papers on the epitranscriptome as well, to put another label on it, and the effects of that. And so I suppose, epigenetics is an umbrella term. It would cover the modifications, chromatin structure, long coding RNAs. We choose to look at sort of DNA methylation because we kind of see it as the ultimate biomarker in a way. It's on the genomic context. It's changes over time. It's also relatively stable, which is key. So obviously looking at RNA can be a pain.
Did you mention already, I'm sorry if I missed this, but like, do you look at the entire genome, or do you, choose select sites that are correlated based on methylation patterns with certain outcomes?
Right, yeah, no, so across the genome, there are approximating, let's say, around 30 million sites which would be methylated CpG sites that would be grouped into different sort of features and structures. We've highlighted about 5.6 million of the most sort of useful and interesting. So to talk about, I guess, depth of sequencing here, which is, I guess, quite topical. We look at 5.6 million at 30x depth, but cover a whole lot more, over 20 million at sort of low pass sort of depth. And I guess compared to the next sort of array, I guess, analysis you could do, which is 850K sites, this is kind of a massive jump above. And we actually see a lot more sites, for example, in correlating to certain phenotypes of, say, lifestyle risk, for example. We can detect sort of methylation changes across a whole gene rather than what might have been picked up as just a single site previously. So can get much more resolution into the dynamics of that, what's happening over time as well, and with dosage as well, things like that. So it's
very interesting.
Suddenly, it's kind of maybe getting a bit off track here, but just really gives a sort of dynamic look into the genome and makes it active. And
yeah, okay, very interesting. Now, I wonder if, and I think that you touched on this with your introduction, and I think this question goes to both of you, however, how you could describe the patient demographics that would best benefit from using this technology. And this is kind of a two-sided question, meaning, of course, you think everyone's going to benefit, but the repositories have notoriously been full of white European American genomes. And that's been, of course, in the last couple of years, there's been a large push to increase the representation of the genomic material that we have in these repositories. So how will efforts like that by the NHS in the UK help to, increase that representation?
Right, yeah, it's a very, really kind of pertinent point to these days. And I think, I guess the parallel is also in, we see in AI, in various technologies using AI that have biased data sets and resulting outcomes that, can cause outrage in the media, etc. And that sort of effect is, even, I guess more stark in biological sciences because the data sets are even smaller and more expensive. And yeah, I mean, I do hope that the NHS is looking to be very inclusive in its to gather some big chunks of that report we mentioned, which talk about inclusivity of, ethnic backgrounds, obviously, but also disease types, yeah, of course, making sure that what we're able to produce in terms of biomarkers and genomic medicine is relevant to everyone. I think there'll be a double-sided approach to it, where you'll, representative models that have been built on, fully representative populations, and then matching up to the personal genome, your own personal genome, that'll be able to kind of, I guess, correct or provide you with a kind of a more nuanced insight into who you are particularly. But importantly, not miss you out if you don't happen to be white, Caucasian, which is the I guess the majority of the study samples.
Right, and I would imagine having technologies that, like yours, Joe, that rely on having a lot of those genomes in the repositories. It's particularly important that the diversity of humans be represented in these different repositories.
Yeah, absolutely. And I think it's one of the key points about using sequencing instead of things like genotyping arrays is that makes this more feasible. And so traditionally, the way researchers or groups have approached really getting to scale in a new population is the design of a genotyping array, where you have to decide ahead of time which genetic variants you're going to look at, and then you sort of commit to that for 5 years or 10 years. And it makes it very challenging to then take that thing that you've designed for a particular population. And a lot of the historical genotyping arrays were designed for European populations and then moved to help understand a different population. And so with a sequencing technology like low-pass sequencing or whole genome sequencing, you no longer have that problem. So by sequencing, you're actually covering everything. And so there's been some really great work out of the Broad Institute by Alicia Martin, some work that we've been involved in with them, sort of specifically looking at how sequencing technologies can enable basically the rapid scale up of genetic studies in underrepresented populations. And so it's one of the key points of sequencing is that it enables these types of things to go quickly.
Absolutely. And I think there's also a question of how this information gets into someone's hands in order to allow them to make a decision. Should this be something that has a clinician, a healthcare provider, gatekeeper that will receive that information and parlay that into a recommendation? Or is the direct to consumer model, which one could argue is truly democratizing the information, but we've seen how information can get passed around and perhaps misinterpreted by people who might not have the correct background training or, just ability to interpret that, to use that to make a decision for their health. I know, for example, that Gencove previously had a DTC, a direct to consumer arm, and I don't think that's part of your business model anymore, Joe. So can you talk a little bit? Yeah, I recall, I actually used to teach at a community biolab here in Brooklyn. And I remember when Gencove came about and I said, hey, there's this really cool new sequencing technology that, it seems like a great alternative to 23andMe because it's not just these tiny little SNPs, you get so much more information. But then it was no longer available for the folks who were taking that class. So can you talk a little bit about that decision and why it doesn't make sense for Gencove to take that direction?
Yeah, sure. So some of these things about what the right way for technology to get out is a DTC or B2B. Like I'm personally a fan of the DTC type approach, but once you take it out, I think what's been interesting is in these 23andMe type ancestry type approaches, a lot of the key interest is around genealogy and ancestry. And that's been really the biggest businesses in DTC genomics have been built around that. And so that ended up being a very challenging market for us. And so we decided to focus on the strengths.
That makes sense. And it's nice to hear that you are a fan of DTC, because I believe that is the Chronomics business model, and you guys sell a kit that people can, kind of assess their own epigenetic changes over time, even to see how lifestyle changes may affect their epigenome. Correct, Toby?
Yeah, no, interesting you bring that up. So the answer is actually at the moment we are not direct to consumer, although.
Oh, you're not?
I'm sure we could sort something out if people are interested. So please do get in touch. We did start fully direct to consumer, thinking let's go full on, be the epigenetics, the 23andMe of epigenetics, etc. And however you want to define it, we were the first direct to consumer epigenetic company using saliva and sequencing, definitely. And the use of saliva actually is another point. Maybe we should kind of come back to that in terms of accessibility as well. And so I guess there's the accessibility of cost, but also sample type as well. How you
absolutely
samples. But yeah, no. Now we work with clinics mostly, and sort of key partners, could be corporate wellness, etc. But our platform is designed for preventive health, with wellness and the quantification of risk factors in mind. So not diagnostic for cancers, etc. As yet, but that's definitely we're pursuing sort of all angles and applications of it. So yeah, I mean the DC model I think is very powerful. I think there are big questions over, well, not questions, but you want to avoid the miscommunication of data, and I think you want to avoid the spreading of more further anxiety, etc., and unnecessary kind of load on healthcare, which is maybe a sort of slightly paranoid view of it. I'm a massive fan of it, but I think there's going to be a huge opportunity for genetic counseling businesses. I've already seen the US that are kind of growing very, very well, and dare I say, the first epigenetic counseling business as well, to kind of sit between that and kind of ensure between sort of healthcare and the D2C sort of world to ensure that kind of information is used properly. Because used properly, it's incredibly valuable. Obviously, it's just a question of data handling.
Data handling, and I would guess that there's a little bit of education even for clinicians, for them to keep up with the latest data and know what's been validated and what is still to be determined. So do you have any experience in trying to do some of that clinician and healthcare provider education, or do you work mostly with the genetic counselors?
No, I mean, so definitely, with many of the clinicians we work with, many are very interested in genetics themselves and epigenetics as well. But it's not, obviously, they aren't professors in statistical genetics from eminent institutions. So it's not down to them to really kind of understand the nitty-gritty. They got to understand the key thing, which is how does this provide value to my patient journey, and from the patient side of things, how does this increase obviously my chances of getting better, but also how is this kind of analysis that I'm paying for, my payer is paying for, outcome, and that's kind of what is the main sort of communication piece to doctors will be, is how do you use this to better your patients.
Yeah, I see, and I guess that having some sort of case studies or literature study in order to back that up is always helpful, as well as
I guess goes to premise of how does it be not just descriptive but prescriptive in terms of the actual patient journey. So.
That is exactly where I wanted to go next. Thank you, Toby. But I do want to talk first with Joe and ask if you've had any challenges in, the same way that I asked Toby, with people who are scientifically trained, healthcare providers, but don't keep up in the day-to-day, or if they spend their, continuing education credits somewhere else, you might have a little bit of a challenge in getting this into the clinic. Have you had any of those experiences?
Yeah, the way we approach it is generally that when we get have those types of discussions, what it suggests to us is we just need to go one step further, right? So people are asking, how do I interpret it? How do I interpret this? That means we need to build a better interpretation engine that helps them, give some concrete suggestions for how to use these type of data in what they're doing. And I think we're seeing this a lot in genomics, sort of the one of the big academic trends, these things called polygenic risk scores, which are even mentioned in this UK report for really early sort of population scale screening of disease risk. And these are statistical measures of adding up thousands or tens of thousands or hundreds of thousands of genetic variants into some sort of score that says people with this particular score are more likely to get breast cancer or to have heart disease or whatever it is. And we know from a research perspective and a statistical perspective that this matters. This is an important risk factor. But if you try to take this into a clinical approach, the response is often, well, okay, what do I do? Like I got this test, my patient scores the 95th percentile, now what? And I think that gets to the question of exactly how do we take it to what next?
Right. And so let's go into how can we actually make patient decisions, healthcare decisions based on some of these. Of course, knowing your health risk can help you to change your lifestyle, and maybe you could even combine that with some of these epigenetic tests in order to see whether you are decreasing the activity of these different loci around these different loci as you eat healthier or exercise more. But you mentioned a couple at the beginning that were covered in this report as well. Like we've already talked about pharmacogenomics. What are other types of ways that we can take genetic information about our cells in order to change the course of what a medical directive is?
Yeah, so I think oh, go ahead, Toby.
Sure. All right. I'll just mention it. Maybe you can pick up on it as well, because maybe you're thinking the same thing. But a big one would be, for example, in cancer subtype classifying, where you're taking tumor biopsies, you're sequencing it, and you're looking at not only what genetic sequence of that tumor, but also increasingly what's the methylation profile. And yeah, I mean, the approach of precision medicine in oncology is absolutely going towards, sequence it first, that's becoming routine, and then see what to do after that, because that just kind of makes sense for a genetic disease as cancer is.
Of course, that makes sense. Joe, you were saying?
And I was on the sort of population screening level. There are sort of a handful of sort of genetic risks that can be treated in some way. If it's high risk of heart disease, there are things you can do to affect heart disease risk. And there's now some pretty decent evidence out of some of the studies out of Finland and some other sort of large biobank studies that if you inform people that they're a substantially increased risk of heart disease, it does actually change behavior. So people might be more likely to stop smoking or to exercise more or to change their lifestyle habits if they know that they are personally at increased risk of disease. And so I think there are a handful of these types of examples where letting people know that they're at increased risk of a disease does lead to relatively straightforward sort of non-surgical interventions that can help them.
You mean that the people who learn that they are at risk are more likely to actually undergo those lifestyle changes, which can sometimes not be super fun.
Exactly.
I think I'm remembering a conversation I heard, I think it was Michael Joyner out of the Mayo Clinic, who was saying that we shouldn't rely on precision medicine for this. We should just tell everyone to change their lifestyle habitat or habits and, eat healthier, exercise more. But I think that any study that backs up that genetic information can really
it doesn't land the same, right?
Yeah.
If that's generic advice for everybody, it probably doesn't land the same as like, no, look, on this chart, you're in the 99th percentile. It's right. That's not looking good for you.
Yeah,
absolutely. I think definitely down to human psychology there, we'd all be at a much lower risk of population level of many diseases if we followed, a few basic healthy habits. But I think the fact is if you do see that you have that risk factor within your genome, that it is a powerful motivator. And I say, you know, of course, to always trumpet the epigenetic side of things is being able to actually quantify how those risk factors in the environmental sense are impacting you. So for example, take smoking, for example, you'll have certain predispositions to be vulnerable to the effects of it. You'll also be able to measure literally how much is that impacting you right now, and quantify improvements as you change your behavior. So that kind of like actionable feedback loop, I think is really important as well.
Yeah, absolutely. So I want to move into the future and talk about how some of these technologies might be applied going forward. But we do have a question from the audience, and I don't know, Joe, if this person is teeing it up for you, but the question is, what applications do you see in animal health? And I know that Gencove has recently moved into animal genomics, which can be used in a variety of different applications. Please excuse the background noise. So maybe you could talk a little bit about what those initiatives are.
Yeah, absolutely. And so people in the human genetic space don't often realize this, but there are many millions of genetic tests done per year in animal breeding. So in dairy cattle, in beef cattle, in chicken breeding, and swine breeding, and in corn and soybean and a number of other species. And so this is where we think, so Gencove can make a big difference here, in that this fits in with our mission of really making things scalable, cost-effective, really industrial scale. And so we've been working with a number of companies in the animal space to do exactly that. And so you can imagine the same types of things that you do in human. You can think of 23andMe for cows, right? So a cow is at risk for disease X, is more likely to have children that produce more milk. You know, these types of things are extremely important in the different industries, but require both scale and cost efficiency that something like low-pass sequencing can help provide.
And I would guess that some of those genetic traits are going to be a lot more important than necessarily like ancestry information for animals like cows and
it's also,
hens.
I'd say, yes, and a lot of these sort of quantitative predictions where there's no physician saying like, what does it matter if my cow is at 3x risk for heart disease? It's someone managing the entire population that says, well, I'm going to breed this cow with that cow, and I'm going to actually change the population mean of milk production or disease risk or whatever it is. And so it's an industry that is very advanced and thoughtful about these sort of high-level statistical approaches.
And do you find working in the animal health space that some of the interventions or I guess implementation of tests for things are a little bit easier simply because it is safer to experiment or try new things in the animal health space than in the human health space?
Yeah, absolutely. There's still consequences to getting it wrong, but it's true. It's not quite the same. Yeah.
Absolutely, there's consequences. So this is another question from the audience, and thank you very much to those of you who are attending and sending these in. And I think that probably you can both talk on this, and this is, what are your thoughts on figuring out what specifically exercise does in order to change the body, and use that in order to make a drug that might target those same, to get that same effect? Does that seem like a good use or a possible use of some of these technologies that we've talked about, perhaps to get something that might mimic the effects of exercise on those same genetic loci?
Absolutely, yeah. So just really quickly though, on the previous question, I would mention, so
please, I'm sorry, Toby, yeah.
Genetic biomarkers for animals, for the animal world is not totally out of the question as well. It's something that's kind of come up very recently. Is sort of an interesting, new development, which is, so we'll see how that goes. And for example, what's the biological age of, your pet, for example?
Oh, sure, yeah.
So using exercise interventions as a proxy, I mean, absolutely. So in the engaging longevity world, there is a lot of research on the effects of dietary restriction as the most effective lifespan extender as an intervention. And many, drugs and sort of drug candidates currently even in the big name pharma are looking at sort of mimicking or targeting the pathways involved in that. For example, take metformin, for example, or drugs targeting the IGF signaling pathway. Yeah, definitely in the case of exercise, that would be a very good thing to do. I can't think off the top of my head of any kind of studies targeting particular pathways involved in that, but the answer is yes, that's a very good idea. I think.
Can you talk a little bit more, Toby, about how you use epigenetics in cancer screening? You've talked on that at the very beginning. And is that a place that Chronomics is at right now, or is that something that you have planned for the future?
So that's definitely something we're very interested in. I would say in terms of, that's something we're not doing now, so we won't be able to go in depth in that. What we really, I guess, going back to the sort of the founding sort of, principles of Chronomics was taking a step way back from, diagnosing cancer, screening cancer. Before that, before you even got it, what are the risk factors that end up causing cancer within a few years' time? Let's put numbers on those so you can reduce those now, and so it's not even a problem, so you don't have to think about it. So that's why we focus on those risk factors, such as quantifying, the effects of smoking, pollution, alcohol, metabolic risk and things like that, and aging itself, of course. In terms of looking at different cancers, for example, one example of the application of epigenetics is in brain tumor classification. So using DNA methylation is currently the most accurate way of classifying different brain tumors. And in many ways, we say cancer is an epigenetic disease. If you think about as even more often you get changes, you have changes of many genes within a tumor that are potential drug targets for pharma companies, but also opportunities for precision screening and more accurate treatment pathways. So yeah, it's definitely a huge area. And in terms of, I guess, as a snapshot of what's, I guess, coming in, maybe give a view from the pharma world, is that there are 7 epigenetic drugs currently on market, mostly targeting cancers, processes within cancer. In the pipeline, there are well over 100, potentially 150. So it's a massive sort of area that's coming essentially.
Very interesting. Yes, I think there's a lot of applications for the future. And now I would like to maybe take a trip to the future where we're talking about some of the challenges that may have been addressed, right? So we're talking not only about different applications for perhaps tumor classification, but you mentioned earlier, Toby, that even sample collection might be one of the challenges, or what do you see, not necessarily within the technical field, although you're welcome to go there, but challenges in getting this to be, let's say, part and parcel of your annual physical checkup, to have your epigenome or your low pass whole genome read so that you are able to use that information as you continue. I know right now, I have not used either of your products, right? I would love to. I think that would be great. But how do we incorporate that into our healthcare system?
Right, absolutely. Yeah. So I guess my view of that would be, in terms of genetics and doing a whole genome, I'd love that to get to a stage where that's just at birth, everyone's sequenced, and you'd have, that's your genetic passport you have with you, take it to the doctor or whatever. And then it's again, for example, if you're looking at a tumor, unfortunately, whatever. But over and above genomics, there is the whole world of the actionable omics, proteomics, epigenomics, metabolomics, etc., that gives you the snapshot of who you are as you develop through life, and how we're going to get those samples in a repeated fashion. That's kind of, that makes eventually prevention becoming the unthinking norm in a way. So yeah, I mean, there's a lot of new technologies coming out in terms of what's possible. Blood microsampling devices that take very little, even some non-invasive, semi-invasive, I call it, skin patches, needles. We champion saliva as a tissue of choice, and I guess a lot of people are still amazed how much we can do with it. In terms of genomics, obviously you have leukocytes and epithelial cells, so everything's there. And there's certainly a lot of innovation sort of opportunity for even say appliance companies to get into incorporating genetic sequences or other sampling technologies into why not a toothbrush or your toilet, for example, or so we have the omics universe coming to you, and you not having to get an awkward kit and send it off in the post and wait a few weeks and that kind of thing. Another factor in that will be the speed of these technologies as well. I think the one big, I guess, step change will be so we'll be, I don't know, I'm going to call it the real-time omics evolution, which might come, and that would be quite interesting if that were ever to happen. What if you could get these results much faster? And we're seeing those kind of innovations with COVID testing driving the sort of rapid molecular diagnostics. So why not the Star Trek tricorder to take it real in the future?
Absolutely. Wow, that is, I mean, it's a lot to bring into the healthcare system, but I agree, like even allowing people to have a daily, monitored event and not necessarily making it annual, that would certainly give people a lot of information to make their daily decisions on. Joe, what do you think about bringing low-pass genomics into more common usage? Do you see this being something that is like you go to a specialist, you work with a genetic counselor in order to make sure that you understand and interpret all of the information, or where do you think the future is going in this sense?
So I think it's been one of these interesting things to be in the field for a while and talk about how genomics is going to get into healthcare. I think we're now at the point that it's not really a technological problem per se anymore. It's sort of an incentives and willpower problem, and how do we actually show the benefit to a healthcare system. And so I think if I had to say what would really move the needle, it would be, at least in the United States type insurance and healthcare systems, it would be these big health economic studies that say we've screened hundreds of thousands of people in our health system. 3% of them had a particular type of disease that we were able to catch earlier, and that's important medically, it's important scientifically, but it also saved the health system X millions of dollars over 5 years. And I think that's the type of thing now that would really move the needle. I think the technology is there, the ability to interpret data is there. It's the problem's now at a different level of just willpower and showing the clear value to health systems, at least in the US. And I think the UK, it might be a slightly different situation.
Maybe a question for Joe there, just in terms of what you see in terms of, as you mentioned, health system adoption. I mean the NHS is fantastic if it gets going. It is a big beast there, and it's kind of like a tanker in terms of where it can go. But in the US, you obviously the kind of structure is potentially more flexible, and you have these great examples. Like one I keep going back to is Geisinger Health System and Regeneron partnership, and I'm just wondering if you see more health systems in the US becoming like that and very proactive, or potentially other parts of the world, Singapore, places like that. Or...
In the US, I would say absolutely. The Geisinger type approach, I think, really showed a lot of people that this was possible, and a number of progressive health systems are now thinking along those lines. It really has to be sort of one of these health systems where everything's kind of integrated with the insurer and the provider sort of working together. And so it's kind of these special cases like the Geisingers of the world, as far as I've seen. But that example has really thrown into relief like what is possible in the U.S. healthcare system.
Excellent. Very, very interesting and grand ideas for how this will be incorporated going forward. Now, you said that the technology is more or less solved, Joe. Can you just let us know how much it costs to sequence a low-pass genome, a low-pass sequencing genome?
So our list price for a low-pass whole genome for humans is $50. We do things where we incorporate sort of key actionable genes. So this is sort of the ACMG 59, where it's not just low-pass whole genome, it's low-pass whole genome plus BRCA, plus the LDL receptor plus these important actionable genes, that's slightly more expensive. But it's that's the ballpark that we're talking about.
Yeah, so it's not really a cost of the technology. It's more about
Yeah, no.
resource allocation by using these population studies, and then governments can say, well, we should put more money into preventative care in this sense or that.
Exactly.
Okay. Very, very interesting. Now we're nearing the top of the hour. And so I guess what I would really like from each of you is your wish that if the general public had this knowledge about genetics and genomics, what is the one clear message that you wish everyone on the street understood about how we can use this information in our healthcare system? I know that's kind of a complex question to ask, but this is a very complicated topic that not everyone delves into on a daily basis, and yet is becoming part of our daily lives and the way that we live. So how would you distill that into a message that hopefully everybody would be able to understand? And I'm going to give you like maybe 10 seconds before I call on one of you, because I didn't share this question with you, but I think it's an important one.
Yeah, I mean, I'd say that the genome is equally or more important than things like family history and the sort of standard things that we care about in the health system now. Like the genetic interpretation is, in most cases, far surpassed that, and is more important.
I think that's something that everyone can understand. And when I say everyone, I usually use my brother and sister as examples. Smart people, but not biologists. Toby, what do you wish that maybe my brother and sister would understand about genomics and health?
I'd say just to kind of have a think about how your genome is, and every single trillion or trillions of copies of your genome in your body is fantastic, dynamic 3D architecture that kind of controls such an intricate processes that isn't just a static string of letters, and that's obviously incorporating a lot of molecular biology in there, but I think that's kind of the next step in terms of really kind of appreciating what's going on down there. And I guess as an outcome, potentially it's encouraging more and more people to go out and have their genome sequence through low pass or other technologies or whatever, and to get involved in research as well, and to kind of really kind of appreciate that they can give back and help sort of build this future as well.
Yes, I like that call to action for everyone to be a part of the future, of our genomic future. Thank you so much. Joe, Toby, this has been, for me, a whole lot of fun. I hope that you also enjoyed it, and I hope that everyone out in our audience also enjoyed. Don't forget to join us again next week. We will be talking about telemedicine and clinical trials and how those two are intertwined. I would like to thank you, Joe, for spending time this hour with us to share your experience and your wisdom.
Thanks for having me. Really appreciate it.
And thank you so much, Toby, for not only sharing your wisdom, but sharing it at such a late hour in such a cogent manner, so much more cogently than I would be at 10, I think 10-ish p.m. your time, right?
That was an absolute pleasure.
Yes, wonderful, wonderful. So thanks everyone for attending our event, and we look forward to seeing you again next time.